Hello there and welcome to Botany 101. My name is Angela Nishimoto. Okay, so today we're going to go into plant breeding and propagation part 2. Okay, we're going to talk about plant propagation or continue to talk about plant propagation. So if you remember last time we were talking about reproduction, right? We're talking about sexual reproduction, whether it's self-pollination or cross-pollination. We're talking about asexual reproduction, whether it was through crown division or through cuttings, whether leaf cuttings or stem cuttings. And we're also talking about tip layering and air layering. Well, think to yourself, what if you had this tree in your yard? You have this tree in your yard, you go out in the morning, you pick a grapefruit and you have that as part of your breakfast from the tree. Okay, you're happy there eating your grapefruit, right? It's good and it's juicy and it's something that you really like for breakfast, you know, your vitamin C. And then for lunch, you go out and you pick an orange from the same tree, okay? And this is sort of like your little snack after lunch or your dessert after lunch if you're good and eat fruits for dessert instead of other sweets, okay? And then for dinner, you go out and pick a lemon from the same tree, okay? And then you use the lemon to squeeze onto some salmon that you're cooking for your dinner. Okay, you're broiling some salmon, you put a little salt on it, a little dill, and you squeeze lemon on it after it's done. Okay, you might see this is impossible, but that's not necessarily so. Okay, because there's something called grafting. Okay, grafting happens when you have pieces of different plants connected to each other. Okay, so this illustration here is showing a cleft graft. Okay, there's a cleft cut into this bottom section. Okay, and a different plant branch is actually inserted into the cleft. It is actually cut into like a wedge shape to fit in here. Okay, and once a graft is done, and if it's successful, both the parts will grow together as one plant. Okay. So people have been doing grafting from at least 1560 BCE or before the Common Era, or 1560 BC, okay? And this is a very useful technique, but in this way you can clone, the clone plants might be hard to propagate via cuttings, okay? So you can actually get the clone and put it on a different plant and propagate it that way, okay? So many fruit and nut trees are propagated this way. Think of mango, think of lychee, think of apple, think of different citruses, okay? So what these are done is that they're bred for high quality products such as fruits or nuts, okay? And these branches are grafted onto a part called a rootstock or a stalk, okay? They're grafted onto strong stalk. So the part called the scion is the top part. Scion means son or offspring. Okay, so you have the stalk, which is usually very hardy, okay, very resistant to insect pests and diseases. And then you have the scion, which actually has desirable fruits or nuts. Okay, so with grafting, you can actually graft grapefruit and orange and lemon on citrus stalk, just like that example we were talking about a couple of minutes ago. Okay, you can also graft almond, plum, and apricot on peach stalk. Okay, amazingly so. So you can actually have, you know, 3 or 4 kinds of fruit on one plant. Okay, you can also have apple trees with 2 or more varieties on the same stalk. Okay, and of course many of you might have seen some novelty cactuses that are red or yellow in color. Okay, they don't have chlorophyll, that's very apparent. So what they'll do is they'll graft the colorful cactus on a regular green cactus. And of course the green cactus is doing photosynthesis, moving sugar into the other part. Okay, so what you need between the stalk and the scion, you need good contact between the vascular cambiums of the stalk and scion. So sometimes you cut both at an angle, or you can do a cleft graft like I was showing you before. Cut them both at the same angle and make sure there's a good connection between the vascular cambiums of the scion and the stalk, and then you can actually maybe get a successful graft. Okay? Now another way of propagation is to specialize stems and roots. Okay, so you have bulbs, like for tulips, there are natural propagules of tulips. Okay, corms, as in taro or kalo, Colocasia esculenta. Okay, you have the tuberous roots of sweet potato or Ipomea batatas. Okay, and you have rhizomes, such as in ginger, Zingiber officinale, okay, the edible ginger. Okay, now typically these are fleshy, okay, with food storage tissues, and they have buds, okay, that sprout, making stems. Okay, now another way of doing asexual reproduction is through micropropagation. Now, a major drawback of most asexual propagation is that they also propagate pathogens. So once a plant is diseased, that's it. But then in micropropagation, you can actually get disease-free plants. So it's possible if you grow them in a sterile environment, because usually when you're growing plants in a pot or plants in the ground, they're exposed to pests, whether they're aphids or mealybugs or nematodes or any kind of animal or other kind of fungus, you know, any kind of pathogens that might get on it. So if you actually grow them in a sterile environment, they stay clean, but you need to start with clean stock with beginning, okay? So you need to get disease-free plants. So another thing about micropropagation, you can actually get a large number of plants in a very small area, okay? So that's another advantage, is also that you don't have to do very much maintenance in it, because plants that you're growing in the ground or in a pot, you need to water them, you need to fertilize them, you need to repot them or reapply fertilizer every so often. But what they do is they do rapid multiplication in culture, okay? And what they'll do is it's similar to growing cuttings, okay? So you're growing them in vitro in test tubes in a sterile medium. Okay, and the medium is usually made of agar. Remember, we talked about agar before. It's a product of red algae. Okay, you have salts that stand for the nutrients, the macronutrients, and the micronutrients. And do you remember that mnemonic for the macronutrients and the micronutrients? You remember that? C. Hopkins Cafe, mighty good, not always clean. Come in, cousin Moby. So you need to remember that, okay, that particular mnemonic, okay? They also include sucrose as a energy source in the media, okay? Also vitamins and hormones are necessary. Then they adjust the pH, okay? And then pour them into test tubes. Then they autoclave it. Autoclaving something is like putting something in a pressure cooker. It's actually heated under pressure in order to kill any and all microbes that might be in it. They'll kill almost all of them. Sometimes it's impossible to kill every single one, but they pretty much do most of it, okay? And this medium provides support for the plant in the test tube, okay? Also supplies nutrients and water, okay? And these tissue cultures are maintained in a controlled environment, usually with artificial light and controlled temperatures, okay? So micropropagation happens in sterile conditions, and they rely on totipotency, okay? This is the ability of like a single cell to actually give rise to any tissue in the body. Okay, they're like human stem cells, like marrow stem cells in plants, okay? So they begin as explants. So what are explants? They're excised pieces of root or stem or leaf, okay? So you must remember that every cell in a plant body has all the genetic information that it needs to make all parts of the plant, okay? You just have to know how to turn genes on and turn genes off, or actually encourage growth of roots or growth of stems, depending on what you're doing, okay? So in terms of tissue culture, there are 4 steps. Okay, first of all, okay, tissue culture, okay? You need to disinfest the plant material, okay, to remove surface contamination without killing the plant, okay? And what they'll usually use is bleach or ethanol. When I worked in tissue culture, I actually used to work what they call working in the box. Okay, we used to use Clorox. Okay, so It's a disinfectant. Okay. Then the plant part is inserted into the test tube onto the media. Okay, and that's usually done in a box or a laminar flow hood. It's like a lighted area with a seat at it where you can actually do the tissue culture, and that'll it actually flows air through it toward you to keep from anything you're saying or anything you're bringing into the lab getting into the plants. Okay, that kind of lessens the amount of contamination. Like a usual rate of contamination when you're doing tissue culture work is about 10%. That's an average contamination rate, and that's a pretty good rate. Okay, so what you do is after you insert them into test tubes and cap it and sterilize it, then you get sterile, viable plant tissue culture. Usually sterilizing it, you flame it in a Bunsen burner, okay, like the tip or the cover of the test tube or the test tube cover, okay. Now the second step is you develop multiple shoots. How do you do that? The microshoots are separated and put in new medium. That's called self-culturing. Okay, and during this second step, you can get thousands of plants, okay, within just months, okay, and it depends on your hormones that you put in there, how long you leave it in there, how often you propagate it, okay. Now number 3 is called root formation, okay. The plants are transferred to a rooting medium, okay, so that's a agar plus and hormones. So what they do is they increase auxin. Remember auxins, okay, that we talked about before? They actually allow for the growth of adventitious roots, okay? So increasing auxins and decreasing cytokinins. Cytokinins, you remember, allow for tissue differentiation, right? So they're oftentimes used to initiate stems, okay? And then 4th, you transfer it to the outdoors. So within the test tube, humidity tends to be high, okay, because it's capped and it's in a sealed environment, right? It's in a flask or in a test tube and it's capped, right? So it's very humid in there. So generally plants that are in test tubes, they don't actually develop as thick a cuticle as you would as if they were growing normally, okay? Another thing about them too is that their stomates don't close as readily when exposed to heat stress or water stress when they don't have enough water. So they need to be acclimatized. Okay, what does that mean? That means that they're actually put in a growth chamber with reduced humidity. Okay, then they're transferred into pots. Sometimes they're put into community pots, also known as compots, where you have a few to many plants in one pot. That kind of helps keep the moisture level up, and they'll keep them usually in a special shade house for that as the plants harden. Okay, and while they're in these pots, they maintain the high humidity to make sure that they get a good survival rate. Okay, they usually use it to harden the plants and get them ready so you can put them into bigger pots. Okay. Now, commercial micropropagation is successful with plants that propagate slowly asexually. Okay, they can be quickly increased. You can get many plants. Okay, orchids, which tend to take a long time to grow. That's one of the reasons why they're so expensive even now. You get a plant in a 2-inch pot, you go to an orchid show, you might be charged $5 for it. Okay, Boston fern and another plant that tends to propagate slowly if you divide it. Okay, African violet, you can get it from the leaf cuttings, of course, and hosta, another plant that tends to be slowly propagated asexually. Okay, you can quickly increase them. You can actually get as many plants produced in a month as would be produced in a year or even more, because you can actually make thousands of orchid plants, okay, using tissue culture or cloning. Okay, you can get new cultivars multiplied to meet high demand, because new cultivars, it tends to be like timely, like fashionable. Like one kind of plant might be fashionable at one time, and people want this plant at this particular time. So what they do is they actually multiply and clone it and multiply it to meet the demand for it. Okay, so one kind of plants that are in high demand is new apple stalks. Remember, stalks are the rootstocks that people use to do grafting with. Now, new apple stalks are in high demand because other asexual propagation methods can't supply enough apple stalks to actually make it in the trade. They need to propagate them through tissue culture because it's faster. Okay. Now, tissue culture is also used for endangered species. Okay. You can actually make small pieces of a few plants, a few valuable and rare plants, and make an unlimited number of plants. Okay. So there's a place called the Lion Arboretum. Okay. So hopefully we'll go there. Yeah, to the Lion Arboretum. And they have a tissue culture lab there. All right. And this tissue culture lab has been responsible for saving at least 5, possibly more, endangered species from complete extinction. These are native Hawaiian endemic plants that they save from extinction just by tissue culturing them, propagating them, because then if you keep them in a test tube, they can stay small. So just as long as you change the medium or change their test tubes every once in a while, you can keep the plant forever small, you know, for as long as you can, okay, until you're ready to plant them out in the environment. Okay? So this ends our little view on plant breeding and plant propagation. Okay? So now we're going to turn our attention to evolution. All right? Okay, here we go. Okay, so the outline of topics for evolution include evolution itself, okay, the first, the second, and the third revolution of evolution, okay. We're going to talk about Charles Darwin, right, the author of the theory of evolution, okay, evidence for evolution, microevolution, okay, and maybe macroevolution and polyploidy, okay. Now, first of all, the author of the theory of evolution was Charles Darwin. It just seemed to be in the air at the time that he lived. He was born in 1809, okay. At the age of 50, in 1859, he published a seminal work called On the Origin of Species by Means of Natural Selection, okay? And in it, he outlined his theory of natural selection being a driving force for evolution, okay? So in this, he stated that organisms that had favorable characteristics that were suited to their environment have a tendency to survive and reproduce new generation, okay? So On the Origin of Species had a great impact on society in general and biology in particular, okay? So evolution means change over time, okay? And here's a look at the book, The Origin of Species. This is probably a first edition, okay? This is the book that actually made Darwin at the top of the most 1,000 influential people the last 1,000 years, something like that. Okay, he was #1 on the Times list at the turning of the millennium. Okay, so organic evolution is accumulated genetic changes in populations over generations, because evolution doesn't happen to individual organisms. It happens to populations of organisms, or groups living in populations in a particular place at a particular time. A particular species living in a particular place at a particular time. Okay, so evolution means change over time, and organic evolution is genetic changes that accumulate in populations over generations. Okay? So evolutionary biology is a fairly new field, of course, okay, only since the early 1900s, okay, or even in the mid-1900s. So with evolutionary biology, this field, you can understand organisms from the molecular level, from its DNA, okay, all the way to the ecosystem level. So an ecosystem is like a rainforest ecosystem, is a physical place, okay, with all the abiotic or non-living parts of it, and all the biotic or living parts to it. So all the organisms in a particular place at a particular time, and the physical environment. That's an ecosystem. Okay? Now, evolution can actually explains the unity of life. Okay? And by studying common ancestors, can actually explain common ancestry, because in evolution, we believe that all living things all stem from a single ancestor, including bacteria, including protistans, including fungi, including animals, including plants. Okay? Evolution also explains the diversity of life, okay, by considering adaptive change to organisms, or populations, to populations. Okay? So evolution is the single most important concept in biology today. Okay? It affects all these areas of science: medicine, agriculture, forensics, biotechnology, and economics, and even law. Okay? So here's looking at a family tree of the living species, or the certain living things that live now. Their names are here, but they're too small to see. But here's actually supposed to be where the common ancestor to all living things is placed in this family tree. And all living things actually stem from two branches. Okay, and then you see that they branch off into more and more branches. Okay. Anyway, so this is just kind of showing you how complicated descent is, but still, it's a simple idea. Well, it's a complex idea and a simple idea at the same time. That all these living things, all of them originated from one common ancestor. And we'll talk about the evidence for that in a little bit. Okay? Now, evolution in agriculture. Okay, so we've done artificial selection. We've talked about this before during breeding. So humans have done this for thousands of years. Okay, we have changed plants and animals to suit our needs. Okay, so here, okay, you have teosinte. Okay, the ancestor of modern day corn. Look how different they look. And this is a hybrid between teosinte and corn. But they're still so similar to each other that they can actually produce a fertile hybrid. Okay? So this change in plants and animals is evolution as directed by human beings. Okay? So we depend on our domestic plants and animals that we've bred and selected from their wild ancestors. Okay? So without artificial selection, we would still be hunter-gatherers. Okay? So we depend on domestic plants and animals for food and clothing and other things that we've talked about. Okay? So here's, remember, teosinte, corn, and their hybrids. See how different they look, teosinte and corn, but they're still closely enough related that they're actually very similar to each other genetically. Okay, just the expression of the genes are different. Okay, but they're very similar. Okay? Now, evolution in agriculture. So agriculture is also the agent of change. Okay? It can influence the evolution of native species. Okay? So we know that insects can develop a resistance to insecticides. Okay, so what happens is that the insecticide or selection will kill most of the insects that you spray. Okay? But some will survive. Okay? And the ones that survive have genes that allow them to survive. And what they tend to do is they will mate and they will tend to pass on these genes that allow them to survive insecticide. Okay? So you get insecticide resistance. Okay? So what you have is that just a few generations it takes for the population to evolve resistance to insecticides. Okay, so people who study insects, also known as entomologists, they try to delay pesticide resistance in insects. Okay, they might actually use or advise alternative insecticides, or look to alternate insecticides using different chemical families for killing insects. Okay, they also encourage the establishment of populations of natural enemies of the pests. See? So you can actually go and buy ladybugs or lady beetles, you know. These are carnivores, and what they'll do is they'll eat aphids, they'll eat mealybugs, you know, other insects that suck sugar out of plants. Okay? You can actually encourage the natural enemies of pests instead of spraying an insecticide. Okay? Insecticides are toxic. That's why they kill insects. Okay? The evolutionary medicine is a new applied field. Okay, an applied field is a field that has practical uses. What it does, it applies evolutionary principles to the treatment of disease or illness. Okay? So of course we know that bacteria infect humans. Okay? And we also, most of us know that if you get sick and you don't use your antibiotics that are prescribed for you properly, okay, new strains of bacteria can evolve. Okay? So the thing is, the bacteria already have the resistance to antibiotics, but if you don't use antibiotics properly, that makes it more likely for the ones that can survive to actually pass on their genes to new generations. Even though bacteria don't do sex, they actually have other ways of propagating. They divide through fission. Okay? So some strains of bacteria that exist today are resistant to all antibiotics, and this includes MRSA, which is methicillin-resistant Staphylococcus aureus, MRSA, MRSA. Okay, we have MRSA in Hawaii. All right, they actually have found MRSA in waters off Ala Moana Beach Park. Okay, there's methicillin-resistant Staphylococcus aureus. There are deaths in local hospitals due to infection by this particular strain of Staphylococcus. Okay, that man that you might have heard about who fell or was pushed into the Alawai Canal, he actually died of MRSA. That's what he died of. Okay, he didn't drown, he didn't get beat up or anything like that that killed him, but it was actually bacteria that killed him. Okay, also there are strains of tuberculosis that are like extremely drug-resistant tuberculosis. I actually knew somebody who died of tuberculosis. He was in the Peace Corps in Kenya, and they think he probably got it there. But I remember when I worked for him, he was my boss, that when I worked for him, he would cough kind of deeply, you know, and then years later I heard he was sick. And I was in school at the time, so I didn't have time to visit him in the hospital, and then next thing I knew that he had passed away, you know. So there are new strains of bacteria that are resistant to all antibiotics. Okay, dangerous, right? Now there are products that are produced, okay, by plants and animals that are natural compounds that can be useful for medicines. Okay, so the World Health Organization, or WHO, actually states that there are more than 20,000 different plants have been used by humans for medicines. Okay, and so what they'll do is they'll send out expeditions to actually harvest plants from the wild, from people's gardens, and grind them and find them. They'll actually grind up the plants, isolate some active principles, test them against cancer cells in particular, but other cells too. Okay, so this is the way they discovered the Pacific yew, which is a source of Taxol, which is an anti-cancer drug. And Taxol is used to treat hard-to-treat cancers such as ovarian cancer and some breast cancers. Okay. There's also the rosy periwinkle of Madagascar. Also another cancer drug is derived from that. Okay. So here's some practical applications for evolution. Okay, because other plant products are used for the smell of it, or as emulsifiers, which means that they can actually hold fats and water together. Emulsifiers have one end of them bonding to fat, another part of them bonding to water. Okay, so one part is polar, one part is non-polar. Okay, other food additives are also gotten from plants. But we can sometimes predict where we'll get a new source of a drug by looking at relatives of plants that supply drugs that we already know about. Okay? Now, species extinctions and invasions, okay, and this is a problem, a big problem, especially in Hawaii, okay? Now, because of our human activities, species have been disappearing at an alarming rate all around the world, but especially in biological hotspots, okay? And this, of course, means a loss of natural products that maybe could have been useful to humanity, okay? This loss of ecosystems too also undermine ecosystems, so you can have this burning or clear cutting of tropical forests to make for farmland, because of course there are more people. We've already established earlier in the semester there are already too many people on the planet, okay? So these ecosystems actually depend on complex interaction among their members, among their different organisms that live in the ecosystems. Okay, so evolutionary biologists study species invasions, okay? So these newly introduced species oftentimes establish at the expense of native species, okay? So you have plants introduced where they have no predators, okay? So instead of making compounds or making physical structures in order to ward off predators, they can put all their energy into reproduction. So this is a look at, they expend their energy in reproduction, okay? So they establish and outcompete natives. So this is kudzu, okay? It's a real rampaging vine. It can grow up to 65 feet a year, choking out native vegetation, especially infamous in the southeastern part of the United States, okay? In Hawaii, of course, we have other noxious pests. We have pests such as strawberry guava, okay? One of my professors actually was taking another professor from another country on a walk, on like a little nature hike in one of the easy trails around Honolulu. And this scientist said, he was Brazilian, he said, well, what is this plant, growing over here? It doesn't look familiar to me. It looks kind of familiar, but I can't identify it. And my professor said, well, it's from your own country. It's native to your own country. It's strawberry guava, Psidium catalanum, okay? And the professor from Brazil said, it doesn't grow this way in Brazil, because in Brazil, this particular plant, strawberry guava, actually has natural predators. They have insects, they have fungi, they have bacteria that prey on strawberry guava. Okay, but here, of course, it's not native to Hawaii, so you didn't have predators or insects or fungi or anything else that evolved along with it. So if you go hiking like along Aya Loop Trail or along the trails around Tantalus like that, you might see stands, whole stands of just strawberry guava, nothing else. Because what they'll do is they'll actually grow by themselves, and other plants can't establish under them because they grow so thick, or they might actually have an effect on plants around them that prevent other plants from germinating, okay? So that's actually the danger of transplanting a plant from one place, especially from a continental area, especially to an island like Hawaii, okay? So these species extinctions and invasions are especially problematic in Hawaii, okay? So the invasive species have been especially apparent, okay? Now, especially because Hawaii is the most isolated group of high islands in the world, okay? You have foreign, from ships and planes, you have foreign plants and animals, fungi and microbes. Here's a cookie frog, I think it is, okay? So what they'll do is they have a tendency to outcompete or prey upon native vegetation, okay? So we in Hawaii have 47 of the 100 most invasive species in the world, okay? They compete with natives for space, okay, crowding out natives. And of course, the native species are nowhere else, okay? The native endemic species are plants or animals that live only in Hawaii and nowhere else in the world, so there's nowhere else they can live. If they go extinct here, they go extinct all over, and they never come back. No organism that has ever gone extinct has ever come back, okay? So we need to understand the interactions between natives and invaders, and if need be, we can intervene and maybe help the natives. Okay? It's actually like a battle cry, a rallying cry of Hawaiian botany, okay, in helping native endemic plants survive and hopefully thrive in this ever-changing environment, okay, which is a hard thing. Okay? Now, about biotechnology and evolution. So the biotechnology industry actually is very interested in the fate of transgenes. Okay? So we talked about this before, it's about GMOs, genetically modified organisms. So some GMOs have herbicide-resistant genes. Okay, so what that allows farmers to do, it allows farmers to apply the herbicide and not to damage crops, because it'll kill all the other plants, but it won't kill the herbicide-resistant crops that have herbicide-resistant genes. Okay, but what happens when the weedy relative lives nearby and may mate with a crop plant? Okay, probably big trouble. So there are consequences to everything we do. For every good thing that happens, there might be a consequence that might not be so good. Okay. Now, in terms of early development of evolutionary thought, okay, more than 2,300 years ago, there was Aristotle. Okay, what he did is he arranged all known organisms from the simplest to the most complex in what he called the scale of nature. Okay, and he implied that organisms are static. That means that they do not evolve. Okay. And of course, you probably know that Aristotle's ideas dominated the Western world at least until the not medieval, but the Renaissance times. Okay, until the 1700s. Okay. Now, fossils had been found for centuries, but the people thought they were like remnants of giants or remnants of the Titans, you know, those creatures of mythology, Greek mythology, Roman mythology. But they weren't identified until the 1400s when Leonardo da Vinci, the genius, okay, stated that there were parts of organisms that were extinct, that no longer existed. So he correctly stated that fossils are actually parts of organisms that were extinct, or remains of organisms that were extinct. Okay? So in the 1700s, Comte de Buffon, Georges Louis Leclerc, okay, he actually showed that there was evidence that organisms change through generations. So it wasn't until the 1700s somebody actually came out and said that maybe organisms change. Because remember, we also have the Judeo-Christian view of things, that living things were created, right, one by one, okay? So in the 1700s, Georges Buffon. Also, there's evidence that organisms change, and in the 1700s to the 1800s, Georges Cuvier used comparative anatomy to classify animals, okay? But even though there was some advancement in this one, he firmly believed that organisms didn't change, okay? Now, Jean-Baptiste Lamarck was a nobleman in the 1800s, okay? And he was a prominent supporter of the idea that changes in populations over time did happen, but it was due to acquired traits, such as the giraffe's long neck, okay? So he sort of stated that the giraffe's long neck came about, maybe the giraffe started off with a kind of short neck, okay? And as the giraffe actually stretched its neck to reach up toward more leaves, the neck stretched a little bit, and this acquired trait was passed down to the next generation of giraffes that stretched their necks a little bit more, and then they passed that down to the next generation, so then you have the long neck of the giraffe. So Jean-Baptiste Lamarck is maybe mocked for that, but he was actually among the first prominent people to say that species change. Okay? Now, early development of evolutionary thought includes the thought of epigenesis. And this is actually something fairly new in terms of knowledge, okay? And it actually does allow for the inheritance of acquired traits, not in the way that Lamarck said, because this was not due to mutation, Okay, what happens is that you have the blocked expression of genes by the addition of methyl groups to DNA. Okay? So if you remove the methyl groups, then the expression of the gene is restored. Okay? So mRNA expression can also be controlled by methylation. And here's a look at methylation, okay, or epigenetic mechanisms, as it says here. Okay, so they're affected from different factors and processes. So it can happen during development in utero or while the organism's, being in its womb. Okay, if it's an animal. Okay, or in plants if it's still in a seed condition. Or you have environmental chemicals can actually stimulate this methylation. Or drugs or pharmaceuticals might do it too. Or just natural aging or your diet can actually affect this. Okay, so what happens is that a methyl group, okay, here actually latches on to a piece of DNA. Okay, now these blue things are proteins called histones. The DNA wraps around. Okay, so this area that the methyl group attached to is now inaccessible. This part of the DNA is inaccessible, so the gene that it actually has on it is inactive, rendered inactive by the presence of the methyl group. Okay, so this actually can lead to some health problems such as cancers, such as autoimmune disorders, such as mental disorders, and also diabetes. So this is kind of interesting kind of thought and very important, of course, So epigenetic phenomenon might actually be responsible for a lot of those conditions. Okay. So these epigenetic changes occur during development of the organism. Okay. And oftentimes it's reset during sexual reproduction. Okay. Except there's an exception. But in corn, these changes are carried through generations. Okay, so they think the reason for it is it allows for short-term adaptations. Okay, they can possibly reverse phenotypes. Okay, so you have these organisms like corn, they can switch phenotypes or the physical expression of the genotypes. Okay, and that depends on the environment that the organism is in. So isn't that fascinating, epigenesis, right? That's definitely an area of study for the future. Okay. Now we're going to talk about the revolutions in evolutionary thought. Okay, the first revolution. Okay, in 1831, Charles Darwin set sail on the HMS Beagle for a 5-year voyage. Okay, he went on the Beagle. There's a picture of the Beagle. Okay, as a ship's naturalist. Okay, so after he returned, after 5 years, well, he hiked in the rainforest. He looked for fossils and discovered fossils. He, of course, famously went to the Galapagos Islands. And when he returned, he wrote about his discoveries for about 40 years. And after about 20 years, he didn't actually start writing on the origin of species until about 1856. Okay, but after about 20 years, he was forced to publish, okay, on the origin of species by means of natural selection. Now, the publication of this book affected not just biology, but affected psychology, anthropology, even arts and literature. Okay, it actually gave us a new perspective of what it means to be human. Okay. So Darwin's travels allowed documentation of evidence. Traveled along South America. We'll go into that a little bit later. Okay. So he described natural selection as a mechanism for evolution. Okay, to support the fact that evolution happens. Okay. So that's the first revolution. The second revolution, remember Mendel? Do you remember Mendel's work? Okay, here we have a monohybrid cross. White flowers, red flowers, all the offspring have red flowers. all the offspring are heterozygous. You have one allele for white flowers, one allele for red flowers, all of them. Okay? Then you self or sib this F1 generation. Okay, and then when you allow them to breed, and allow them to have offspring, you'll get 1 to 2 to 1 genotypic ratio, and you'll get a 3 to 1 phenotypic ratio, or 3 red-flowered organisms for every white-flowered organism. Okay, remember that? Right? So in the 1930s, there was a new burgeoning field called population genetics. Okay, so one of the things you have to remember is that in 1900, people still didn't know about chromosomes. They didn't know about meiosis or mitosis. They didn't know that there were genes. Okay, so in the early 1900s, they found out that genes were on chromosomes. So that started this field of population genetics, which studies about how genes flow through populations and generations. Okay, so here's a look at the peppered moth. Okay, this is a good example of evolution in animals. Okay, so you have the regular form of the peppered moth, which tends to, you know, like most moths, they're active at night and they rest during the day. And during the day, the peppered moth rests on tree bark, which happens to be dappled gray, so it's its perfect camouflage. Now, during the Industrial Revolution, you had a lot of soot, you know, from the smokestacks of the factories. So you had a lot of black stuff on the trees. So the black mutant, which did occur naturally, got to be more populous. There was a bigger proportion of this dark mutant as opposed to the regular wild type. Okay. Then when they actually started pollution controls and the, you know, the soot got cleared away from the trees, then the population of the regular wild type peppered moth actually predominated. Okay, so they actually have that documented as a kind of evolution. Okay, very good example. Okay, so then what happened during the second revolution was that the theories of Darwin, okay, natural selection, Darwinian natural selection, Mendelian genetics, and population genetics all intersected and made the great hybrid, or made the great intersection. Okay? Now the third revolution is what they call Evo-Devo, or evolution of development. Okay? So DNA sequences show that different organisms actually share the same genes. Okay? So the mouse and human genome are very similar to each other. So why do we look so different? Okay? They're actually genes called homeoboxes. They're actually regulatory genes that act as developmental switches. Okay? They're like master control genes. They turn other genes on or turn them off. Okay? So the homeobox genes turn on the right gene at the right times, which leads to proper development. Okay? So proteins are made at a specific place and time. Okay? Here's looking at the different genes for different segments of the fruit fly, Drosophila melanogaster. Okay, so here is a gene that codes for this mouth part. Okay, of course, fruit flies are arthropods. They have jointed feet or jointed legs. Okay, they also have an exoskeleton. Okay, they include insects like this fruit fly. Here's another gene that codes for this part. You remember earlier I told you that they had actually had a mutant fruit fly that had legs growing out of its head instead of antenna. Okay, so all these different genes code for the various parts. You could actually bombard the fruit fly eggs or fruit fly larvae with X-rays and induce mutations in it. So this is why people don't want you to get too many X-rays. They're dangerous. It's high energy radiation. Okay? Anyway, so for the same genes can be used for different purposes. okay, with these homeoboxes. So there's no need to reinvent the wheel every time, okay? Because the interesting thing is master switches of plants are similar to the master switches of animals, amazingly enough, okay? So the example we looked at earlier, teosinte, they have tiny, hard kernels on a very small ear, okay? So human beings actually selected for of a master control gene, and it changed the ear development into corn. Okay, so here's teosinte, here's corn, and here's a hybrid that can form between them. So they're very similar to each other. Okay? All right, let's see. So Charles Darwin himself, when he was a little boy, here he is as an older man, he showed an early interest in natural history, but deferring to his father's wishes, because his father was an MD or a physician. He went to medical school in Edinburgh. Okay, he was really revolted though at the barbarity of surgery at the time, just sawing off people's legs and things like that. Okay, so he instead attended Cambridge University. He transferred to Cambridge and majored in theology. What is theology? Theology is studies about God, about religion, right? So he graduated in theology in 1831. And just at that time, the HMS Beagle was commissioned by King William IV for an around-the-world voyage. And the mission of the HMS Beagle was to chart coastlines, especially the coastline of South America. Okay, so Darwin became the assistant naturalist and captain's companion. Okay, this is Charles Lyell, because he took Charles Lyell's book with him, and this book was based on ideas by John Hutton that all geological earth changes come from very slow processes. Okay, so you have the erosion of rock and debris, making ocean sediments, and sediments accumulate, becoming sedimentary rock, and then the land then is raised above sea level to become land, terrestrial land. Okay, so he postulated that actually Earth was very old, much older than people thought, because people at the time thought that Earth was only about a few thousand years old, maybe 4,000 to 10,000 years old. Okay, so this gave idea that accumulated small changes lead to big changes. Okay, so that was a book that probably influenced Darwin very much. So this is a book he took with him when he was on the HMS Beagle. Okay, so when he was on his trip, he collected plants and animals. He was seasick a lot of the time. Okay, so he spent as much time as he could on land. He went to South America, the Galapagos Islands, Australia, New Zealand. It was like a world tour, like a lifetime adventure. And he also kept a daily journal of what he did and what he saw. Okay, so in 1836, he returned to England. He worked on his plant and animal collections and on his journal. He also carried on correspondences with biologists. Okay. Also on his own, he investigated pollination mechanism, earthworm biology, okay, biogeography, or where different organisms come from and why, etc. Okay. And he was also guided by the essay he read on human populations and food supplies by Thomas Malthus. Okay. So Thomas Malthus's main point was that the human populations grow until food supplies dwindled, there's disease, okay, there's wars, and all these things limit populations. Okay. So maybe Charles Darwin might have thought that wild organisms are also vulnerable to selection in nature, although we ourselves might be able to increase our food supply by selective breeding that we talked about earlier, and also cultivation. So wild organisms too might be vulnerable to selection in nature, okay, which would explain changes in natural populations. Okay, so Alfred Russel Wallace sent an essay in 1858. So Darwin started writing his On the Origin of Species in 1856. Okay, here's a picture of Alfred Russel Wallace, sentiment essay, talking about natural selection being the reason for the origin of new species. So Darwin thought, all my originality shall be smashed. Okay, so at the behest of a mutual friend, Darwin and Wallace made a joint presentation to the Linnean Society, and in 1859, Darwin published On the Origin of Species. Okay, so that's all that we have time for today. We're going to stop here. Okay, we'll continue with evolution next time. And until then, aloha.